Okay, hello guys. Today we are going to talk about the Guyton and Hall, chapter 2, the cell and its functions. So let's start. So the first thing that Guyton talks about is organization of the cell. So in this blue topic, it emphasizes 5 important things, which are the components of the protoplasm. Protoplasm means basically everything inside the cell. You can call it protoplasm. So I wrote it down here, and you can see. So protoplasm consists of 5 important things: water, electrolytes, proteins, lipids, and carbohydrates. And I'm going to talk about each of them, why they are important in protoplasm and why they are constitutes of the protoplasm. Of course, in water, 70 to 80% of the cell is water, and all the chemical reactions takes place in water solutions. That's of course thing. And the second thing is electrolytes. There are so many important ions, for example, calcium, magnesium, phosphates inside the cell, outside the cell, which basically control the cellular mechanism. And third is proteins. If we talk about the proteins, we have structural and the functional proteins. In structural proteins, we have phospholipids. That's not a protein, but we have integral protein and peripheral protein in cell membranes, right? And about functional proteins, we have enzymes. And fourth one is lipids. Lipids is triglycerides, phospholipids, and cholesterol. If we mention lipids, there are so many lipids. And one of them is, I wrote down 3 examples: triglyceride, which is 1 glycerol molecule and 3 fatty acids, and phospholipids, the building block of the cell membrane, and cholesterols, the main component of the steroid hormones, right? Right. Okay. Next, carbohydrates. So if you talk about the carbohydrates, carbohydrates acts as a marker of the cell, every cell I mean. I will talk about it later, but carbohydrates are glycoproteins, acts like glycoproteins, and they locate in the cell membrane. Okay, next let's talk about the cell structure. So some organelles need to have membrane for their functions because they do have special functions inside their organelles. For example, nucleus contain a DNA, so it must separate itself from the cytoplasm. That's the one reason. And there are some organelles which has a membrane also. If we say that cell membrane is one protection and organelles has protection from the cytoplasm also. And cell membrane, nuclear membrane, endoplasmic reticulum, smooth and rough endoplasmic reticulum, and mitochondria, and lysosomes and Golgi apparatus. These has membranes. Okay, next. Now let's talk about the cell membrane. So, cell membrane basically separates the cell and from the environment. And cell membrane basically consists of phospholipid bilayer. Now, what is phospholipid bilayer? Okay, you know the meaning of bilayer, but what is phospholipid? If you break down the phospholipid, it consists of phospho plus lipid, means one phosphor group and one lipid, right? So if we see the phospholipid structure, it consists of hydrophilic head and hydrophobic tail, and together they will make the phospholipid. You can see glycerol molecule here, and you can see the head here, and you can see tail. If you see thoroughly, you can observe two tails, but one of the tail is a little bit to the right because of fatty acid structure, specifically cis structure, right? Now, next, water-soluble molecules can't enter inside of the membrane due to lack of interaction with nonpolar tail. So there is a saying in English that says that likes solves likes. So which means that only polar molecules dissolve in polar solvents. Non-polar solvents dissolves in non-polar solvents only. Because, for example, let's say that water-soluble molecules. So what's the definition of dissolving? So if I talk about the process of dissolving water molecule, you know the water molecule, right? HOH, and it has hydrogen bonding actually. So if it attracts well with the oxygen molecule, it can dissolve. Okay? Now I'm going to talk about the components of the cell membrane, except phospholipids. So we can say that sphingolipids, cholesterol, integral protein, peripheral protein, carrier protein, carbohydrate, so many. But you might wondering, what is the sphingolipids? So sphingolipid is basically like phospholipid, but the structure is a little bit different. But it acts barely same. So in the phospholipid, the basic form or the skeleton is glycerol, but in sphingolipid, the skeleton is sphingolene, the molecule called sphingolene. And sphingolipids is the cell membrane of the nerve cells. You can find substantially in nerve cells, especially the myelin sheath. And the next is cholesterol. So what's the function of cholesterol? Cholesterol's function is fluidity of the cell, and it is the basic component of the steroid hormone. I mean the starting material. And the next thing is integral protein. If I mention about integral protein, it's just, you can just say that it's ion channel for the transporting molecule. In Guyton, it says that in the chapter 3, it will explain thoroughly about the integral protein and peripheral protein. Not the peripheral protein, the integral protein. And the next is peripheral proteins. Peripheral proteins are the receptors. You can just basically remember like that. And the carrier protein. So carrier protein is just special type of ion channel. And carbohydrate. Carbohydrate is, as I said before, it's the marker of the cell. And there is a term called glycocalyx, which is just the carbohydrate code. And in the Guyton, it was written that glycocalyx has so many functions. For example, it helps for the attachment to the other cell, and because it has a negative charge on it. The carbohydrate has basically a negative charge, so that's why. Next, endoplasmic reticulum. So I tried my best to draw the endoplasmic reticulum. So if you see it clearly, you can just understand that nuclear membrane is double membrane. There is inside a nuclear membrane, and there is also outside nuclear membrane. And endoplasmic reticulum is the continuous of the outer one. Yes. So this is perinuclear space, the space between the outer and the inner nuclear membrane. And we also say that foldings basically cisternia, which is it means that just foldings. So in the Guyton, it clearly said that perinuclear space, in other words, the inner nuclear membrane, and outer nuclear membrane space, and the matrix of endoplasmic reticulum is interconnected with each other. So in the endoplasmic reticulum, you have two types of endoplasmic reticulum. The innermost endoplasmic reticulum is called rough or granular endoplasmic reticulum, and the outermost is called smooth endoplasmic reticulum. The difference is that only the ribosome is there on the rough endoplasmic reticulum. And also their functions are different. For example, smooth endoplasmic reticulum is responsible for the lipid production, and rough endoplasmic reticulum is responsible for the protein formation. So you might know that before, you might know it, but there is a system or pathway, you can say that. So there is one substance which is formed from the endoplasmic reticulum, and it is sent to the Golgi apparatus and then packed there and modified there. But in the Guyton, it emphasized that no. In endoplasmic reticulum, there are also some substances which can be synthesized there only. I will explain it later. Now let's go to Golgi apparatus, third one. So Golgi apparatus, as I said before, which is really close to the endoplasmic reticulum because their functions are interconnected. And its real function is it packs and modifies substances that formed in the endoplasmic reticulum. For example, lysosomes, which contain the enzyme called hydrolases. Hydrolases is just basically you're breaking down the molecules using a water molecule and secretory vesicles and so on. However, Golgi apparatus to synthesize alone, for example, okay, let me see. Yes, Golgi apparatus synthesize hyaluronic acid and chondroitin sulfate. In the Guyton, it emphasizes the hyaluronic acid and chondroitin sulfate, their usage. For example, they are the major component of the proteoglycans and major component of ground substance and etc. But I don't want to talk about very deep things. I just want to explain the main flow of the book. And in the lysosome, as I said before, lysosome has some enzymes, right? And that enzyme breaks down the protein molecule into amino acid and glycogen into glucose and lipid to fatty acid. You must remember that. I think that is really important. Next. So there is also an organelle called peroxisome, which is really like lysosome, but in terms of structure and the function, there are two important differences between the lysosome and peroxisome. One difference is in lysosome, which is formed from the endoplasmic and Golgi apparatus pathway. We in the Guyton it is said that we believe that peroxisome is self-replicative, or it is formed from the smooth endoplasmic reticulum, but it is which is still unknown. And the next difference is that in lysosome it contains hydrolases, but in peroxisome it contains oxidases, which is basically H2O2 in chemical composition. And the fifth one is mitochondria. Mitochondria is powerhouse of the cell, which you have heard it so many times, maybe 100 times. So if you talk about the mitochondria, this time I'm going to just explain the structure of the mitochondria. Mitochondria is self-replicative and it has its own DNA, and it has double membrane like the nucleus. I think that is obvious thing because whenever it has something DNA, it has double membrane, right? So inner membrane and outer membrane, as you might expect. And outer membrane is just protective, and the inner membrane is also called cisternia because it is basically foldings. And inside it's called the matrix, you can see from here. So sixth one is cell cytoskeleton. In the Guyton, it explained that filaments and microtubule, and I searched it for myself. What is filaments? So filaments, if you talk about filaments, in filaments there are three types of filaments in terms of their size: active filament, intermediate filament, and myosin filament. As you can see, it depends on their size. Active filament is the smallest one, and the intermediate is, of course, the average one, and then biggest one is myosin filament. And their also function is also different. Active filament is for the movement, and there's the part of the cytoskeleton. Intermediate is also part of the cytoskeleton, and myosin filament is responsible for the muscle contraction, and we will talk about it later in the Guyton chapter 9 or something like that. And actin filament is also known as the microfilament, and it's responsible for the outer elasticity, as it is generally locates outside or the peripheral side of the cell. And intermediate filament is for it builds the strength and the structural support for the cell. It provides very hard structure for the cell, so that cell will not be so fragile. And microtubule is additionally everything that I said about the intermediate filament. It also provides some transportation throughout the cell. Next, nucleus. So if I have to name the nucleus, in other words, I will name it like control center of the cell, because nucleus basically do two things. It controls every activity inside the cell, and it reproduces the cell. So nucleus has double membrane. As I said before, outer membrane is the continuous of the endoplasmic reticulum, right? And the nucleoli and the formation of the ribosomes. Nucleoli is the plural form of nucleolus. I think you might have heard it before, nucleolus. So nucleolus is just accumulation of rRNA and some proteins in nucleus. And if I just wrote down the steps of how the ribosomes are formed, firstly, we will make the ribosomal RNA in the nucleus. In DNA, in the nucleus will make, in DNA it has some codings, and that will make the ribosomal RNA, and that ribosomal RNA collects in the nucleolus, and that will combine with some proteins and they will make the subunits of ribosome, and that ribosome is immature, and that immature ribosome is sent to the cytoplasm through the pores of the nucleus, and then before the protein synthesis, the immature ribosome will become the mature ribosome. Okay, next, functional system of the cell. So we just talked about some structures of the cell. Now let's talk about the function. So in this topic, I will talk about endocytosis and the synthesis by endoplasmic reticulum and the Golgi apparatus. Okay, so if you say that endocytosis is the ingestion of the cell, we have two types of endocytosis. First one is pinocytosis, which is just basically cell drink. If you drink, you eat smaller, right? And you just eat small particles, for example, protein molecules. And in Guyton, it emphasizes very important point, which is pinocytosis is the only way that macromolecules can enter the cell. I think that's really important one. Here it is written like that. And I just try to draw the pinocytosis mechanism. So basically you have coated pit, and why you might wondering that why it is coated pit, why it is named as coated pit, because it has receptors on it and it is pit. And beneath that, below that, we have clathrin molecule, which is just a filament. And I just draw some proteins here. And if you try to engulf that protein, firstly it will act with the receptors and then engulf, and then on the last steps, the actin and myosin filaments will close the door like that. You can clearly see from here. And phagocytosis is like pinocytosis, but there are a little bit different changes or differences. For example, only few cells can do phagocytosis. For example, white cells can do phagocytosis after the opsonization of the bacteria, right? And I wrote down the steps of the phagocytosis. First is attachment, and then engulfing, and actin contraction, and pinching off. And after the pinching off, you will have the residual body, which is just indigestible substance after the phagocytosis, and then that is excreted by the cell, and that process is called exocytosis. Next, lysosome is responsible for the regression of tissues and autolysis of the damaged cells. Okay, let's talk about its meaning. So regression of tissues is the tissue is becoming smaller. So why it's becoming smaller? So there are some cases that you need to become smaller after certain activities. For example, after the pregnancy or after the delivery, you have to regress the tissue and make it smaller. The uterus is becoming smaller, right? And so autolysis is basically meaning this autophagy. I think they are kind of same. So if the cell damage is small, you can just repair it. But if the cell damage is really too big, and that would be the harmful for the body, so the cell must go suicide or autophagy. Now I'm going to talk about autophagy. So damaged organelle is Autolysis and autophagy is a little bit different. Maybe autolysis is the cell suicide and autophagy is you're eating oneself, right? So firstly, you'll have the damaged organelle, and that will produce the autophagosome, and that will become the autolysosome after it combined with the lysosome. So autophagosome means that after you get the damaged organelle, you will produce a vesicle surrounding that damaged organelle, and that is called as autophagosome. And after that, it have to combine with the lysosome so that it can be removed or break down in the with combining with the lysosome, right? And next, I'm going to talk about endoplasmic reticulum function. So endoplasmic reticulum function, it has two functions, as I said before, protein synthesis and lipid synthesis, right? So protein synthesis, after the production of the protein synthesis, it has basically two pathways. One is the protein is sent to the lumen, and one is it is sent to outside the cell by the secretory vesicle, and that is done with the help of the calcium ion. And next is lipid synthesis, is done by the smooth endoplasmic reticulum. Actually, there is really important fact that smooth endoplasmic reticulum is always growing because it is lipid. Lipid means a cell membrane, cell component, right? Cell organelle component. So you can just literally say that the smooth endoplasmic is like a hair, and you are cutting your hair again and again after it is producing and again, right? And intracellular vesicle replenish all cell membrane, which means that intracellular, after we engulf something by the endocytosis, the cell membrane will become smaller in terms of the volume. So we need something so that we can replenish the cell membrane. Otherwise, the cell membrane will become very small, and like that. The intracellular vesicle is responsible for that. Now let's talk about the mitochondria. So I said that mitochondria is the powerhouse of the cell. So if you talk about energy, ATP is the unit of the energy in cellular actions. For example, 5% of the energy or the total amount of ATP is produced by the process called glycolysis, and the 95% is produced by the Krebs cycle. And in other words, it is also known as a citric acid cycle. And adenosine triphosphate. So what is ATP basically? So basically ATP is nucleotide. So what is nucleotide? You are asking me that, right? So adenosine, ribose, and 3-phosphates, this is the structure of the ATP. And as I said before, ATP is the basic unit of energy in cellular activity. So nucleotide is also monomer of the DNA and RNA, which are the polymer molecules. And nucleotide consists of three things: nitrogenous base, sugar, and one phosphate molecule. Another reason why we are saying that ATP molecule is the one unit of the cellular activity is that we can use it like a money. Because after we use money, it just doesn't disappear, right? So we can use it again and again, like for example ATP to ADP and plus phosphate group, and we can use it vice versa. Next, so in food we take carbohydrate, lipid, protein. For example, the carbohydrate, we break it down into the glucose and then glucose to the pyruvic acid, and the lipid to fatty acid, and protein to amino acid, right? So that all these molecules converted into a molecule called acetylcholine. And that acetylcholine is, it goes through the big process called Krebs cycle. So that one I was talking about before, 95% of the ATP is produced by the Krebs cycle, right? And all these things are totally called the chemi-osmotic mechanism of ATP. Now I'm going to talk about the locomotion of cells. In other words, it means movement of cells, how cells move to other places. Of course, there are cells that doesn't move, but there are some cells that can move. So first type is amoeboid movement. Amoeboid movement is basically just like amoeb movement. So in our body, white cells can move. For example, it will become the tissue macrophage, and our cancer cells can also move. And it is called as metastasis if the cancer cell moves. And there are really important term, which is the pseudopodium, which means that the false feet. Through the false feet, the amoeboid-like cells move. For example, I just wrote down the mechanism of how it moves. So from the one side, the toward side, I would say that we will do the exocytosis, and in the back we will do the endocytosis. So I just wrote down the 4 basic steps of how the mechanisms of the amoeboid locomotion is done. Firstly, protrusion, yes. So actin molecule polymerize, and pseudopodium is formed there. And attachment of the receptors is done by the exocytosis, and the cytoplasm will flow into the exocytotic part. And then the fourth one is actin contraction. Yes. Next. So amoeboid. So how the cell is deciding where to go, right? So we can control the amoeboid locomotion by the chemotaxis, which is the process of the control of amoeboid locomotion. And there is a term called chemotactic substance, which is basically the place or the substance that our cell wants to move there. And if that cell is moving toward the chemotactic substance, we will call that positive chemotaxis. And if that cell is moving away from the chemotactic substance, we will call that negative chemotaxis. Next, cell also can move through the process called ciliary movement. So in ciliary, we have 2 types of ciliary in our body, which is motile ciliary and non-motile ciliary. So motile ciliary is responsible for the fluid movement, and non-motile ciliary is responsible for the sensory signals. In other words, it is also known which is also known for signaling antennae. And if you see the structure of the ciliary, it is really a wide known structure. We call it axoneme, which is 9+2 structure, which means that 9 molecules or 9 filament is there and two filament is centralized in the cilia, and that two double cilia is interconnected with each other by the union arm. So in Guyton it is said that there are so many things that we don't know about the ciliary, including its mechanism, how it moves. But however, we do know some things, which is ATPase, the union arm causes a crawl movement of the ciliary movement, and next is front. If front part crawls outward and the back part stays stationary, the bending will occur. So that's all. Thank you for listening. See you next time.